and the temperature below, quote, normal, which is today's temperature, during the period between around 30,000 and 130,000 years ago. And you can see there are these real deep dips in temperature, 8, 9, 10 degrees centigrade below present norms. And that translates, for those of you who don't do centigrade, into between 14 and 18 degrees colder on average in the time the Neanderthals lived. And they survived during much of this time because in places where we find them, they are the only human fossil relative or ancestor that we know of. So they survived a very long period of time during this very jagged, jerky kind of climatic oscillation. And the question is this what did them in? Ultimately, the failure to be able to keep up with dramatic changes in the climate. Well, there have been two stories to finish up about the fate of Neanderthals. One suggests that they, in fact, with a little bit of evolutionary change, became us. That they developed the cultural habits and ultimately the anatomical structures we see in ourselves today. The other, in contrast, suggests that they didn't make it. And that either due to some kind of inability to thrive in a climate that was oscillating over very short periods of time, millennial timescales, simply could not keep up with the ups and downs of their food resources, or that more modern appearing populations, as exemplified by that skull that I showed you, 110,000 from the Middle East, which began to come into Europe by about 50 to 45,000 years ago, displaced the Neanderthals. Or some combination. Maybe the Neanderthals were on their way out. Modern humans, quote unquote, coming from Africa and the Middle East, come into Europe, basically wind up finishing the job. We don't know. The fossil record is mute on the causes of the disappearance of the Neanderthals. And that's where the genes come in. I hope.
I don't know if I can say why they disappeared, I'm afraid. Let's see. Okay, and now to switch gears a little bit and see if I can not fall over the wires. So I'm going to tell you a little bit about what we know about Neanderthal genetics, but first I want to talk about how hard it is, because it's really hard. So ancient DNA is probably the worst thing in terms of genetics that you can hope to work with. And if you enjoy beating your head against the wall, ancient DNA is for you. Okay. All right. So a little bit about dealing with DNA that is old and degraded and horrible. There are a number of challenges with ancient DNA, but the major ones are that after death, degradation begins for DNA molecules and all the molecules in the body. The DNA begins to get damaged, and this causes, so first, the DNA begins to fall apart into small pieces. So this is actually an micrograph of a piece of DNA. Here's a long piece, and then there's some little short pieces. So we deal with a lot of little short pieces of DNA. And it often has chemical damage. And what that does really is cause the DNA to get sort of knotted up and kinked and really difficult to work with. And so that's another challenge to trying to look at this DNA. And then the final issue is contamination. When you are trying to work with ancient DNA, if even a small piece of dander falls in the people who work with ancient DNA get really paranoid, okay? And so we work in clean room conditions. This is actually the Ancient DNA lab at ASU, which is a class 10 HEPA filtered clean room. So it cleans down to virus level particles out of the air. It's positive pressure, so the air is constantly blowing out of that room. So it enters through filters in the top and blows out. And then we end up garbing up in these suits, and this is actually me as a cover girl. Not that you can tell. And in the ancient lab, I actually usually wear more than this. Okay, so we usually have face shields on and things of that nature. So we have to go through a lot of steps to make sure that our results are real, that what we're looking at is really the ancient DNA, that we haven't amplified ourselves. Now that's easy if you're working with an extinct species of non-primate, okay? There are ways to get around it, but when you're dealing with a closely related hominin, that's tricky because you've got to be sure that you're not amplifying yourself. Now, the other thing that I should say about working with ancient DNA is just to sort of, this is the biology 101 part of the talk. You actually have two kinds of DNA in your cells. You have your nuclear DNA, half of which you get from mom and half of which you get from dad. But you also have mitochondrial DNA. And the mitochondria are sort of the power plants of the cell. There's several hundred copies in each cell, and they have their own DNA. And you get all of your mitochondria from your mom, because in the big fat egg, that comes with all the mitochondria and all the stuff, and the sperm basically just delivers the half of the nucleus, or the half of the nuclear DNA. So now, because there's several hundred copies per cell, there's a lot more of the mitochondrial DNA. So most ancient DNA research has actually focused on mitochondrial DNA as opposed to nuclear DNA. Now, Neanderthal genetics really started in 1996. And in 1996, this individual, who is the type specimen for Neanderthal, the Neanderthals, he's an individual that was found in the Neander Valley in Germany, and is known as Feldhofer 1 or Neanderthal 1. They decided to try to get DNA out of this individual. Now you'll notice he's kind of dark brown and shiny here in terms of the bones. That's because they were nicely shellacked back in the 1800s. And that actually wasn't a terrible thing because it probably helped kind of seal off the bones from contamination since then. So we could take off all that outer stuff and look at the time, mitochondrial DNA. So this is when the technology that we had for looking at DNA was still was good. It was much better than it had been, but compared to today, it's amazing how fast the field has gone. So we looked at a short region of the mitochondrial genome that is highly variable. And it was a really interesting region to look at because we have a lot of data from modern humans, and it's been used for a number of population studies. So having a Neanderthal sequence would be really interesting. So what we found from that short fragment, which in total was about 500 base pairs in length, and what we did, by the way, was amplify short little segments of it. So remember, ancient DNA is degraded in small pieces, so we had to use something in biology called PCR, which is really the Xerox machine of the biology world. But we amplified really short 50 to 100 base pair fragments overlapping so that we could put together the whole sequence. And when we did that, we found a very interesting thing. We found that when you looked at different world populations, they were all roughly equally distant from Neanderthal. So one hypothesis was that if they were our direct ancestors, that because they lived in Europe, Europeans should be more closely related to them. But we don't find that in terms of the mitochondrial DNA. So here you can see the Neanderthal is outside of Africa, and they look a lot like every other person outside of Africa. But you pick two Africans, and they can look really different from each other in terms of the variation that we see within humans. Okay, so then people started, so what we said at the time was, it looks like Neanderthals are outside. We think that they were not our ancestors. They were cousins, and there probably wasn't interbreeding. But we said, we're just looking at the mitochondrial DNA, so we're only seeing part of the story. And so a number of other labs started looking at other Neanderthal specimens, ranging in age from 38,000 years ago to even 70,000 years ago. And they extracted mitochondrial DNA from those individuals. And what they found is that the Neanderthals all clustered together. And then here's the range we see in modern humans. And then somebody did a couple of Cro-Magnon from Italy, who are anatomically modern humans. And they cluster right there with us, where the Neanderthals are outside. So difference in terms of the variation. Now, they look really different here, but just to give you a sense of just how different are they, it's within the range of variation that you see within chimpanzees as a whole species. Okay, chimpanzees today are divided into 3 to 4 subspecies, and the variation between Neanderthals and modern humans fits within that. Okay, so I still wouldn't necessarily call them a separate species. They do fit within this idea of perhaps a subspecies. But our results also suggested that they weren't interbreeding. So here's another family tree based on the additional data, and then here's chimp and bonobo as outgroups, just to give you a sense. So this is the range of variation in modern humans. Here's the Neanderthals, but you don't see them intermixed in terms of mitochondrial DNA. Okay, so that was kind of the last word around 2000, 2002. Neanderthals, modern humans, probably didn't interbreed. Then Svante Pääbo's lab at the Max Planck Institute for Evolutionary Anthropology decided that this new sequencing technology meant that, hey, let's go for the genome. Everybody was like, wow, that will be really cool if you can do it. It took a long time, and it's a lot of work, but they've managed to do a pretty good first draft. And I'll explain what I mean by first draft in a minute. But so what they did was they tested a bunch of Neanderthal bones to try to figure out which ones are going to have the most DNA in them, and also have the lowest amount of contamination. And based on the mitochondrial DNA work that had been done previously, they knew which changes in the mitochondrial DNA were characteristic of Neanderthals and not of modern humans. And using those differences, they could test a bone and say, well, too many paleoanthropologists, they'll have been handling this one. It's 20% of the DNA is modern human, so we're not going to use this bone, right? And then they actually started excavating some of the sites